Hello, and welcome to our Knowledge Culture webinar. Today, we'll be focusing on addressing new challenges for large volume mRNA manufacturing. My name is Stephen Hume, and I'll be your host. Before we move on to the main presentations, let me introduce you to the webinar console. The navigation bar at the bottom of your screen has several options that I'll run through with you just now. And if you click on any of these buttons, they open up and close some of the windows that you have on your screen so that you can personalize your view of the webinar. So button number 1 is the media player, and that allows you to see our presenters. Button number 2 shows you the live view of the slides. Button number 3 controls the Q&A box. We will be having a question and answers session at the end of the webinar. So button number 3 controls that box. As you're viewing the presentations, please put any questions you have for our presenters into that Q&A box. Now, the icon that looks like a question mark is in case you need any technical support during the webinar. So if anything isn't working for you, click on the question mark and we'll try to help. Button number 5 contains the resources for the webinar. So here you can download any documents that we reference or get any further information on certain topics. And button number 6 allows you to register for our next Knowledge Culture webinar, which is about continuous perfusion. Now, of course, we're always interested to hear if you've enjoyed the Knowledge Culture webinars. And so at the end of the webinar, you'll also have access to a survey. But I'll remind you about that again later. We'd be very grateful to hear your comments. So we've got a fantastic agenda for today's webinar. You'll first hear from Kelly Fluke, one of our senior product managers at Thermo Scientific. And Kelly's going to tell you a little bit more about the company and some background on mRNA manufacturing. Then we have our guest speaker, Peter Scheinert, CEO of Amtech GmbH. Peter will be followed by Pirkko Muhonen, a field application scientist specializing in bioproduction here at Thermo Fisher Scientific. And finally, Anna Labrise, field application specialist in purification from Thermo Fisher Scientific. So that just leads me to introduce to you Kelly Fluke, a senior product manager for purification. Over to you, Kelly.
Thanks, Stephen. So I just wanted to get us started with a bit of a background to Thermo Fisher Scientific. So right now, we currently have over 75,000 employees who provide global support and thought leadership across all areas of science. And we really harness the power of all of that knowledge. We have over 5,000 employees within our R&D organizations across the world, as well as over 10,000 employees that are field focused. And what that means is no matter where you are, we can, in normal times, have somebody with you in order to discuss your problems, your challenges, and help you find solutions. And no matter what science you're working on, we're always investing money. Right now, over $1 million invested into R&D in order to make sure that we can help you stay advanced with your innovation. And obviously, strong revenues year after year. We're not going anywhere. And at our core, regardless of our function, our experience, we're all scientists. We're all here to help all of our customers fulfill their scientific purpose, and that's to make the world healthier, cleaner, and safer. And we're always committed to scientific advancement. We take a collaborative approach with our customers to ensure that we can get you an unmatched portfolio across the industry. And really, our depth of capabilities enable you, our customers, enable to push the boundaries of your own innovation. And everything we do is underpinned by quality, and it's a foundation of quality that our products and services are built upon. And our customer value proposition really focuses on accelerating innovation and enhancing the productivity of our customers. We take a strong stance in our partnerships and our collaborations with our customers so we can enable routes to, what happened? So we can enable routes to further development as well. So we have a commitment to quality across the organization, and that customer focus spans the breadth of all of our Thermo Fisher brands. And I'm sure in your scientific travels, you've come across one or more of these brands, and we're continually expanding our capabilities. And really the expansion right now and the focus in manufacturing is really on biomanufacturing, and we're seeing an increase in biologics compared to where we were, say, 10 years ago. And this is really emphasized right now in how we're trying to combat new diseases and things that we haven't been so familiar with. And on that note, if anybody hasn't noticed, we're currently in the middle of a global pandemic, and we're working alongside our customers to fight COVID-19 through the development of diagnostics, treatments, and vaccines. And just to kind of get us started on the mRNA topic, if you look at the vaccines that are in development right now, there's this close to 200 vaccines, either preclinical or in the clinic. And one of the interesting things about the spread of these vaccines is that we normally wouldn't see a lot of RNA therapies so far into the clinic. And this really speaks to the positive effects that we're starting to see, and the growth and the interest in this molecule as a therapeutic.
Thanks very much, Kelly, for that introduction. So just a reminder of the Q&A, if you have any questions that come into your head during the presentations, please just pop them into the Q&A box, which is just below. We're now going to cross live to Hamburg, Germany, for our guest speaker. He's from one of the leading providers of RNA technology products, Amtech GmbH. Please welcome Peter Scheinert.
Yeah, thank you very much, Stephen, and hello from Hamburg. This is Peter Scheinert speaking. I'm CEO and co-founder of Amtech in Hamburg. And here in Hamburg, we are focused on production of synthetic nucleic acids for therapeutic and diagnostic applications. And I'm more than happy to introduce you today in the field of messenger RNAs. The first slide. So, yeah, okay. The question right now is why are synthetic mRNAs so exciting at all? And so we have in former years we had initial approaches for genome therapies using mainly based therapies, vectors, plasmid DNA, and so on. And all of these therapies have at least a risk, even a high risk, due to genomic integration with permanent genetic alteration of cells. And this is definitely something which has created safety and ethical concerns against use of DNA. And so in the last years, the mRNA has become more and more the topic in the focus of research interest. And the major advantage of mRNA is its transient nature, which means that mRNA is only used or consumed also in the cytoplasma. It will not be able to enter the nucleus, and it will, of course, will not be able to generate any kind of genome integration effects. So and also synthetic mRNAs can be produced and can be engineered to function more or less like the cellular mRNAs. And so you can transiently express proteins by structurally resembling natural mRNAs. Of course, for me, there were a lot of concern using mRNAs. Mainly, mRNAs are considered to be very unstable. So overcoming major challenges in this regard of this drug class related to instability, immune activation also, translation efficiency, and also, of course, delivery of the mRNA to cells has proceeded the basis for a wide range of potential applications. And nowadays, mRNA-based therapies are also used in cancer, for cancer treatment, and of course, as we are all aware right now, for vaccine treatments. This is really a very exciting story. And so mRNA enthusiasm has produced really a big wave of emerging companies to further exploit this technology. Next slide, please. You can see that there were some topics to solve at first before mRNA could be really a new class of drug. The first thing is that next slide. Okay. So the first thing was that, of course, improvements of mRNA production have to be made. Cap structures had to be developed, functional cap structures. The poly-A tail is definitely an issue. The stable poly-A tail introduction, because poly-A tail is very important for stable and for functional mRNAs. Also, incorporation of modified nucleotides was very important improvement, because this makes mRNA possible or amenable to introduce into cells without introducing immune response. It's the mRNA itself. And this has led, this was a major impact for accelerating the advance of IVT mRNAs towards the clinic. And only availability of high quality and fully functional synthetic mRNAs has enabled progress of their applications. Production of synthetic mRNAs for application as vaccines, for instance, allows rapid to quickly address outbreaks and rare novel diseases. This is certainly really a very exciting activity of or functionality of this molecule. That's also the reason why we all are aware about that. Currently, many companies are taking great effort to produce worldwide produce COVID-19 vaccine, and this needs also scalable solutions, millions of doses on the mRNA vaccine, and that's indeed also a big challenge right now. Upscaling of mRNA production and purification batches towards, and that is really amazing, from milligram scales towards double-digit gram scales up to kilogram scales. And we did have also requests, we have discussions with our customers in recent times asking already for even kilogram scales of mRNA. This is really incredible, and has of course also impact on the whole production line, supply chain line also. And of course it requires really reliable and very good solutions for upscaling. So generally speaking, we have now reached a very good step, a very good point of the mRNA technology. It has developed out of the early beginnings, where it was considered to be a very critical molecule, now to a reliable, to a very, very promising tool, and to be really a highly interesting new class of drug for the near future. The next slide. Just a little bit about Amtec. So we are located almost in the center of city Hamburg, very nice area, lots of nice restaurants in the surrounding area, hotels, good connection to the airport, railway station, important for our guests, of course. And My name is, as I said before, is Peter Scheinert. From my background, I've studied molecular biology at the University of Kiel, and I was in 1998 co-founder with some other colleagues, also with Guido Krupp, which you can see on the picture as well. I'm on the left side, Guido on the right side. I was co-founder of Artus GmbH. Artus was a very successful story. Artus was a real-time PCR-based company, and it was acquired in 2005 by QIAGEN. And already during the Artus times, Guido and me have been in charge for the development of synthetic nucleic acids for the real-time PCR assays. So I think we have an outstanding experience in production of synthetic RNAs. So we have then, in 2005, after the acquisition of Artus by QIAGEN, founded AMTIC, and this means that, as you can see in the next slide, that we do right now have a real-time track record years. Very exciting years behind us, and looking very much forward to the next 15 years ahead of us with really very exciting times. And we are, of course, absolutely happy and also proud to be part of this exciting mRNA story right now, because we really will have very exciting applications ahead of us, very exciting technology aspects and improvements in new technology developments ahead of us. So we are really very proud to be part of that story. On the next slide, you can see a bit about our product lines. We have two main product lines. The one thing is the diagnostic product line. This is our oldest product line. And the second line is the therapeutic product line. The next slide, you can see that we have listed some key applications at first for the diagnostic product line. So the main application for this product line is used as reference material for development of in vitro diagnostic assays, or therapy companion diagnostics, or companion diagnostic, in other words. Also as internal controls for real-time PCR diagnostic kits. And we were very happy that also during the current COVID times, that the European Commission has reached out to AMTIC in order to manufacture GMP-grade positive control materials, RNAs, to support reliable COVID-19 diagnostics in EU and also worldwide in the later step. So this is certainly for us a good kind of advertisement also, and also good kind of acknowledgement of AMTIC that that EU has reached out to us to support worldwide laboratories being engaged, being involved in COVID-19 diagnostics. And the next slide. Of course, now I think that's the main topic of today, therapeutic mRNAs. So we are able to produce customized therapeutic mRNAs from any sequence up to 30,000 nucleotides approximately. This is so far interesting because at the moment a number of customers are using so-called self-replicating mRNAs for COVID-19 approaches, but also for cancer therapies. Self-replicating RNAs are in the focus, and this means these mRNAs are normally very long, way beyond 6,000-7,000. So it was for us very important to have also the options to produce very long mRNAs. And key applications are, of course, everywhere, omnipresent everywhere right now. Vaccination by an mRNA production, but also tumor therapy, neoantigen therapy, and very exciting also genome editing, and of course protein replacement. And we are also happy in that regard that we are offering customers a fast-track service for gene manufacturing of fully functional SARS-CoV-2 mRNAs for vaccination approaches in cell culture, animal models, preclinical and phase 1 and also clinical trials. This is certainly for us also a very exciting field. And we have also, of course, a lot of requests in that regard. On the next slide, you can see that we were, throughout the years, growing with our customers. Just to give you some information about our customer situation, we are now, we have customers, let's say, we have a broad variety of customers. We have customers, we are international active, means we have customers in the United States, Europe. We have customers from the field, hospital field, but we have also customers from pharma, also big pharma customers who are ordering material at Amtec for their clinical trials or preclinical trials. And you can see that we just to give you some numbers, we have pharma biotech, approximately 14 customers in Europe, same approximately United States and Canada. We have academic customers, hospital, and of course we have also many customers in the IVD branch. We have just visited the biggest one, and also very important for you, for the audience to know that we are already involved in clinical trials with our material. We have the regulatory compliance for that. This is a key factor apart from the quality. We have also regulatory compliance in place, a quality system in place to enable us to enter clinical trials, and we, as you can see, have produced trials for application ex vivo, in vivo, and we are very proud to be also involved in the meantime for clinical trials in patient phase 1-2. We have produced approximately 22 batches right now for that purpose. So this is, I think we are at the cutting edge, and for us it was very important to have that compliance to be able to respond to customer requests asking for this very exciting new molecule of mRNAs for applications as you've seen before. The next slide. A bit about our workflow. I don't want to go too much into detail at this point because it would be certainly too technical. Just to let you know that we have a bit of different flow. We have a PCR-based workflow. In our production, we don't have a plasmid-based workflow. So process starts with a sequence from a customer. We receive a plain sequence. Then we start with the genome, with the gene synthesis step, plasmid preparation step. Then we receive from an external collaborator a circular plasmid DNA. We don't need an encoded poly-A tail in the plasmid. This is a very important aspect, but again, I don't want to focus so much on the technical aspect on this presentation. We are introducing a homopolymeric T120 region by the so-called field PCR. This will be the later poly-A tail, which means that we have a highly homogeneous PCR product for mRNA production for the IVT, which means this is certainly one of our key advantages, that we have a really highly homogeneous template for mRNA preparation with a well-defined poly-A tail lengths and a well-defined TT lengths in the DNA template. And then we have the bulk IVT, of course, and purification of the mRNA product. The next slide, you can see some questions or some thoughts about the challenges. What is the challenges in that field? I've mentioned that already before, that certainly for all key applications we have been I've told you that in the case that the clinical trials are successful or will enter even the market, a tremendous, and recently some customer told me an epic increase of mRNA needs will occur, and this will mean that we have to take care about upscaling solutions, especially for purification, to increase batch sizes from scale. Maybe next question. And there are of course standard methods available, mostly or mainly age purification, but most such methods require multiplication steps and also various chemicals. This is indeed an issue because these chemicals are maybe highly toxic. For instance, acetonitrile. We have X rooms for that special applications. And at the commercial scale, such methodology is on the vaccine scale, where we have really hundreds of millions of doses per annum. It seems to be possible, yeah? So therefore, we are really very, very happy about our collaboration with Thermo Fisher, because we have started our collaboration now half a year ago, or something even longer. And we have seen that the POROS, or the T25 resin, affinity resins, are a really very promising and highly scalable solution for mRNA purification. So I think we have two aspects: scalability, and this is what we have seen throughout all our discussions with Thermo Fisher so far. I really would like to mention as one that we have very productive interaction with the tech support from Thermo Fisher, Germany and US also, both US and Germany. And we have, of course, we are running also a number of proof of concept batches right now, or tests right now. So scalability is really very easy, and also non-toxic. So that's the reason why we want to go for this direction together with Thermo Fisher. The 15P compliant purification of extremely large scales of mRNA, very easy, non-toxic, easy scalable. Next slide. You can see a picture how normally our mRNA looks like. We expect a single peak, a homogeneous single peak. We have a small peak forerunning. This is an internal marker in this picture. So this is what mRNA should look like without aborted products, with any kind of side products. And in the next slide, you can see that it is also very important. Just a moment. No, we have it. Since we are working fully customized, which means that we receive hundreds and even more sequences from customers a year, this means that we see also, of course, sequences which might have high HCC content, invert repeats, tandem repeats, homopolymeric regions, etc., etc. And this will definitely create problems in production of full-size mRNAs. Therefore, it's very important to have a good troubleshoot scenario in place. And you can see in these three examples, we have capillary electrophorograms before and after optimization. Left side, example 1, 2, 3. 3 mRNAs which have, of course, a full-size part, but they have a lot of smear, trailing, non-molecular weight material which would not pass our release criteria. On the right side, you see these mRNAs after optimization. So this is a very important aspect for us, capability for optimization to achieve the best quality for customers' mRNAs. And in that regard also, we see a very big potential and advantage of the porous speeds, because it's an AT selection. The porous DT stretch binds to the poly-A tail of the mRNA, and the mRNA at the 3' end of the mRNA, we would be able to achieve a full-size selection of mRNAs, which means that with the affinity resin, it will enable a first-step purification of poly-A species, which means that aborted products which don't have the poly-A tail will not be purified. So we have a very nice selection of poly-A containing a full-size mRNAs in that purpose, in that context. And the next slide. Just to show you a very nice picture from one of our customers, customer success. So normally, I would really like to point out that we do never ever disclose any customer names. We had a very nice collaboration started already in 2016 together with a Canadian company, Acuitas Therapeutics, and they have conducted, they performed a comparative validation of 3 amplicate batches independently produced according to our PCR workflow versus a plasmid DNA-based workflow. And not going too much into details, although this picture now, but you can see already it's visible that we have the blue, the green, and the red bar, that we have, as Akusis also stated, that we have our expression was approximately 2 more active than current benchmark. It was equivalent to the most active historical commercial batch. And also very important that we had lower plasma cytokine levels compared to standard benchmark. This for us is really very nice and a clear signal about a superior quality of our PCR product as template for mRNA synthesis. And of course, we have, unfortunately, we have lots of other feedbacks like this one, but we will, as I said before, we do not disclose customer names and data unless we have permission to share that. Yeah, the next slide, a bit about our analytical capabilities. So for GMP-grade mRNAs, the list is getting longer and longer. Quality is extremely important because we all have to be aware that in contrast to the diagnostic product line, which will be used only, let's say only, as internal controls for real-time PCR, in this case, the mRNA will be used as a drug and will be injected in humans. That's the reason why, of course, there is really a very high focus also on quality, on purity, and we offer a broad list of QCs internally, but also with external partners. And just to name maybe the most important one of them, we are using the sequence identity check from the incoming plasmid and also from the final mRNA via PCR or RT-PCR. We are checking for residual remaining template DNA because, as we have heard about during the introduction, the advantage of mRNA therapeutics is that there is no DNA present due to the mentioned potential progress of DNA. So many customers are really very concerned, this is for many customers a very important to see, to show that there is really no DNA present at all. And of course, we are performing microbial enumeration tests and bacterial entry testings to show that there are no bacteria or things like that into the mRNA. And the number of additional QCs can be asked from us. And the list is, I think, growing every year. There are coming more and more QCs on the table. This is also kind of a challenge for CMOs like Amtech to react on the requests coming from the customers, but also from authorities in that field. Yeah, this is the end. I would like to thank you very much for your attention. And of course, I'm happy to answer questions which you might have. And then back to Stephen again from my end, from Hamburg.
Peter, thank you so much for that very interesting presentation. And congratulations again on the 15th anniversary of the company as well. If you also found what Peter was saying interesting, then we're running a Science Against SARS-CoV-2 virtual conference on the 6th to the 8th of October. The middle day focuses on vaccine and therapeutic development strategies, and you can find the link in the resources box. So I'd like to introduce one of Thermo Fisher Scientific's field application scientists specializing in bioproduction. Here is Pirkko Muhonen.
So thank you, Stephen, for the introduction, and good afternoon, everyone. So today I will talk about plasmid and recombinant protein microbial production for mRNA synthesis. So when the demand of the mRNA technologies is growing, it will consequently increase the demand of DNA templates and recombinant enzymes, which are the key components in the IVP process. So during the next 10 to 12 minutes, I will go through our Thermo Fisher Scientific solutions for manufacturing and sourcing these molecules. So microbes are commonly used for plasmid or recombinant enzyme production. To be able to grow microbes, any basic media should include nutrition sources like proteins, vitamins, and minerals, some carbohydrates as energy source, usually glucose, and then buffering agents that maintain the optimum pH and osmolality. Peptones and meat extracts are commonly used in microbial broth. For example, standard microbial media like terrific broth TP contains tryptone, yeast extract, glycerol, and potassium salts. So it is important to remember that there is no one fixed formulation for all the fermentation processes, so different microbes have different nutritional needs. So let's talk about peptones more in detail. So these are the common nutrition sources in microbial broth. They are also called hydrochelates and derive from natural sources, for example, yeast, plants, animal materials, which are digested under controlled conditions using chemical or enzymatic processes. The peptones are water soluble and provide high diversity of essential nutrients for microbes as well as mammalian cells. The reason why peptones are so widely used in bioprocessing is highlighted on the pie graph. This graph shows the typical mix of nutritional components that we find in peptones. For example, the amino acids, the vitamins, carbohydrates, minerals, trace elements, all these elements that microbes use for growing and production. They also offer protective effects and enhance performance like increased productivity. So because of the diversity of the peptones, we have specialized analytical testing capabilities so that we can characterize and monitor complex formulations. So on this pie graph, you can see the different analytics we can perform, in addition to the molecular weight distribution, metabolic analysis, nitrogen analysis, and so on. So when the microbe have some of the microbes may have very specific needs in terms of nutrition composition, and this extensive analytical testing can help you to understand the impact of different peptones and optimize the key drivers in your process. So diversity is really the key word when we are describing peptones, and because peptones are digested from different sources, they exhibit different nutritional compositions. So screening different peptones is recommended to identify the exact needs of your particular microbe. So it is important to evaluate a wide range of peptones to identify the ones that really work best in your culture. So to help you to screen and find the best peptone and supplement, we have developed 3 different KIPCO peptone starter packs containing different animal origin or animal origin free or animal origin peptones. These starter packs contain 5 to 6 different peptones, 100 grams of each, and these packs are designed so that you can do fast, small-scale evaluations. And when you find the best peptone or peptones, these are available in GMP grade so that you can source them for your GMP manufacturing. You can download the flyer here to see more details of these starter packs, and we also have a very comprehensive nutrient manual available on our website for more detailed information and data. So the idea of the starter packs is that you can really easily evaluate different types of peptones. And I have now 2 examples to demonstrate why it is so important to test variety of peptones. So on this slide, on the right-hand side, you can see growth profiles of 5 different microbes when cultured in M9 broth, supplemented with glucose and ultrafiltered yeast extract. So as you can see on this graph, this type of yeastolate is compatible with some of the microbes like E. coli, Bacillus, Enterococcus. Sorry, I think we have been moving. So, so on this graph, you can see that the yeastolate is compatible with E. coli and Bacillus and Enterococcus strains, but not so much with the Staphylococcus and Saccharomyces strains. So on the left graph, you can see the distribution of molecular weights after the digestion process. And as I mentioned earlier, that our analytical testing capabilities, the degree of the digestion reflects to the molecular weight distribution and is used one of the process control parameters to monitor batch-to-batch consistency. So here's the second example on why to evaluate different peptones. So in the previous slide, I showed that the E. coli and Bacillus strains both grew well in the yeastolate-based growth media. However, when you grow these microbes in selection of different plant-based peptones, like shown on these graphs, E. coli and Bacillus strains show preferences towards different peptones. And on these experiments, the tryptic soy broth was used as a control medium. You can see it on the blue dashed line. So these results really demonstrate how important it is to screen and titrate different peptones in your cultures to find the most optimal one. So in addition to our wide peptone collection, we have been developing two types of E. coli media prototypes. These are now available for alpha testing. Both prototypes, animal origin-free and chemically defined media, they are easy to use. They are one part of dry powder media. So instead of mixing several different powders to prepare your broth, you can mix only one pack of powder. So these prototypes have no animal components, and therefore the BSE and TSE risk is reduced. And these are designed specifically to support E. coli nutrient requirements. So if you aim for a chemically defined process, the CD E. coli fermentation media is a good fit. And we aim to launch the Kipco-Bacto CD Supreme fermentation production media next year. And the AOF, animal origin-free E. coli fermentation medium, consistently outperforms the terrific broth, and which is animal origin, and we are aiming to launch this in 2022. So if you're interested in testing any of the E. coli prototypes or peptones, please do contact us. So in addition to the microbial media solutions we have, I also wanted to briefly introduce our high-performer single-use fermenters. We have designed these fermenters to fit microbial fermentation requirements. They have direct central mechanical agitation, 3 Russian impellers, 4 baffles to break up the vortex, recirculating condenser, and really powerful mixing to support the high-density microbial cultures. And on this slide, I wanted to show how the high-performer single-use fermenter perform when compared to a stainless steel fermenters. And in this case, P7 RNA polymerase, which is the key polymerase in mRNA synthesis, has been produced in our 30-liter and 300-liter high-performer single-use fermenter in comparison to 100-liter stainless steel reactor. So the results on the SDS page show that the enzyme accumulation is identical between the single-use fermenter and the stainless steel tank. And on the right-hand side, you can see that the enzyme activity is actually higher in cultures harvested from the single-use fermenter. So high-performer single-use fermenters are also used for plasmid production. And on this slide, the 30-liter single-use fermenter has been used for fed batch culture with heat induction. So the plasmid production was induced by heat shock by shifting the culture temperature from 32 degrees to 43 degrees once the optical density of 60 was reached. The culture was held at 42 degrees for 2 hours before harvesting, and these images really show the amount of plasmid in biomass increases due to the increased temperature. I also wanted to mention that this study included the Kipco-Bacto yeast extract and Kipco-Python peptone, and you can now download the application notes on this slide. So in case you don't have resources for your own fermentation process or you wish to progress fast, we have a comprehensive selection of TheraPure IVT reagents. So these are manufactured by using a single-use technology on the right-hand side in the picture. This is the 100% closed system to help to ensure that the entire manufacturing process is never exposed to the surrounding environment and human operators. So here's a polling question. So I'd like to ask the audience to select where or what do you use currently as a DNA template in your IVT process, whether you use a synthetic own production or outsourced, or whether you use a plasmid, again, whether you produce it yourself or outsource it or something else. So I leave this open now for a few seconds and then look at the results. Okay, let's have a look. Okay, it really looks like the plasmids are coming from the own production. Some of them really are outsourced. And yeah, it looks really nice distribution where you are outsourcing your or sourcing your DNA template. So in case that you in case that you want to outsource the DNA or the DNA template product, we have GeneR services that can provide you the high quality plasmid or synthetic DNA that you can then use as a template for your mRNA synthesis process. So GeneR services are really cost effective, and it saves your resources and time if you want to design your plasmid or DNA fragments. And you can easily order online, and please download here the gene synthesis brochure for more details. So I wanted to summarize here. So I wanted to highlight that we have the Thermo Scientific solutions to support your mRNA IVT process. So if you don't have resources for producing your own recombinant enzyme, we have the comprehensive selection of therapy enzymes and reagents. We have the GeneR services for DNA synthesis. And we also have the custom solutions for our products and process liquids if you may need so. If you have your own fermentation process set up, we have the wide range of animal origin-free peptones and also the newly developed E. coli media prototypes. We have equipment such as high-performance single-use fermenters for large-scale production. We also have the capsule select and porous resins for downstream purification. So this is now end of my talk. Thank you for listening, and this goes back to Stephen, please.
So just in case you have already tried to click through to the virtual conference link that's in the resource box, it wasn't working before, but it is now. And if you do struggle with the link at all, it's www.thermofisher.com/science-against-SARS-CoV-2. That's a mouthful. We also have our next Knowledge Culture webinar coming up on the 22nd of October, so you can always register for that webinar as well, where we'll be discussing the implementation and optimization of continuous perfusion. So next up is Anna Labrise, a field application specialist in purification for Thermo Fisher Scientific. Anna, welcome.
Well, thank you, Stefan. So I will just speak now about the purification of the mRNA. That I don't see if the people can see me because I still see you, Stefan. I don't know. Okay. So once you have produced your mRNA, you want to purify it in a fast and efficient way. You may be aware that Thermo Fisher offers two lines of purification: chromatography resin, that the capture select resins, which are mostly based on agarose and are designed for affinity, and the porous chromatography resin, which are based on a porous backbone, which is very rigid and scalable. These porous resin comprise a portfolio of ion exchange, of course, of HIC with very unique features, and lately the oligo DT. All of them are meant to be used in commercial processes and latch test processes, and they are already present in a lot of them around the world. We have a production of poros, which is about 50,000 liters per year, with usually commercial lot size of 250 liters. And for the Capture Select, it's 15,000 liters. Beside the standard offering, we also propose a custom resin development programs to really make a resin for your specific needs. So your specific ligand, your specific molecule. So I will then go in the features of the Poros Oligo DT25 resin that we have launched some months ago now. This is specifically designed to purify poly-A mRNA. So you have here the porous bead, a linker, and a poly-DT ligand, a chain of 25 mer that will bind to the poly-A tail of a poly-A mRNA. All our beads are 50 microns in diameter, meaning that you can reach almost an analytical resolution, but because of the specific technology inside, large bore, rigidity, etc., we can still use them at very large scales, like 500 liter or even 1,000 liter columns. This resin has a very powerful way of, in a simple capture step, purifying quite well the mRNA, which are based on the A and T base pairing. The protocols are not very complicated. You would just use salt to load and elute in buffer without salt or with water. We have reported binding capacity now for an example of 4,000 nucleotide, which is up to 5 milligrams per milliliter. And so far, all our customers report a recovery which is above 90%. The scalability is a very big point for us. As all our porous resin, they are completely scalable. The bead itself can stand very high pressure, can stand very high volume, and we can produce in high volume. It comes with a complete GMP package with a regulatory support file, so the scalability was for us a very important point. And that's the reason why also Amtech, that you saw the presentation just before, chose to go on with this resin. And of course, they are non-animal derived. We have different options or different scenarios to use this resin. So you have the affinity purification, obviously, where you would use this resin to specifically remove anything which has nothing to do with RNA, like DNA, nucleotide, enzymes, buffer, or IVT media components. In principle, anything that doesn't have a poly-A tail would not bind the resin. Then we always, as a second step for final polishing or intermediate polishing, we advise some typical ion exchange chromatography or hydrophobic interaction to remove what could be left there. So basically double-stranded RNA or RNA with different structure like hairpin. And that's another scenario to use after that, again, the same oligo DT resin to, for example, exchange a buffer. So there is different scenario that you can use. We worked closely with Amtech because they had an opportunity here. As you saw, they were a leading RNA CMO, and they had a very much increasing demand for large quantity of mRNA. They needed something efficient, scalable, easy, quick. So we worked with them to develop something that would be an alternative to the typical reverse phase HPLC or the spin columns that cannot be really scaled up. That's why we offered this resin, and we did a lot of tests with their samples, and we came out with the resin that we are proposing now to you, and which is since a couple of months on the market for very straightforward capture step. I come to technical things. So we always advise when you start to work with this resin to first check the tolerance of the salt, to the salt, sorry. from the RNA to the salt. So for example, we tested here NaCl and KCl, the very typical one, and it's very interesting to know exactly what concentration of salt will be tolerated by your mRNA. In that case, you could see that with 1.4 molar, you start to see precipitation, or 1.6 molar precipitation of mRNA, which is not obviously the case with NaCl. This is usually not depending on the size, but it's on the type of the mRNA. It's something that you have to try. It's really empirical, so you cannot really know, depending on the size, the type, the sequence, etc., if it will precipitate or not. Once you know the salt, so we went on with NaCl because it's a more platformable salt. It's quite nice to understand also the impact on the efficiency of the elution. So in that y-axis, you see a percentage of load which is present in the elution pool. So basically everything that you recovered in the elution pool. So when you have 0%, it means that everything was in to the flow through. So you can see here with the two different salts that you could go up to 0.4, 0.5 molar concentration to allow a good binding and a good recovery later on. This is not depending on the size of the mRNA. So what we show here is usually for 2000 nucleotide, but we have data on 1000 and 3000 nucleotide. It is more dependent on the structure, the sequence of the RNA, and for example, a 3000 nucleotide RNA doesn't mean obviously that it will be likely more to precipitate or aggregate because of its size. It doesn't work that way. We just wanted also to check the pH and the phosphate. So we tried with phosphate buffer and trace buffer at different pHs, so 6.6 to 7.8 or around 7.8 and different salt concentration. On this DOE graph, you could see that the best results, the best, in that case, static binding capacity was obtained in trace buffer at a concentration of 0.8 molar NaCl and a pH which is quite classical and neutral. It's very important for your specific process to test all the things to get the best out of the resin. And that's what we can recover. It shows you with a specific load, and we are here underloading, and here we are overloading at 40 milligram per milliliter. How does the recovery and the yield look like? In that case, with 2 milligram per milliliter, we are below the nominal DBC, so we reach actually 95% recovery and 95% yield. We lost almost nothing in the breakthrough. When you are overload above the DBC, you still get 95% recovery. That means that everything that was bound, you can recover. But as we are a bit above the DBC, the yield would be a bit different. You would have a bit of loss in the breakthrough. But that's just to show that you can really play with the condition to get the best out of it. About the recovery, depending on the size of the nucleotide, we didn't see really big differences. So you have the DBC here and the different sizes and what you lost in the breakthrough. Most important here, that's what you recover after the load. So in the elution, we could recover for all the sizes almost 90 to 100%. So the size actually does impact the capacity because, of course, the bigger you have, the less you could bind, and there is spheric hindrances, etc. But the recovery itself is not impacted. So everything that you will bind, you will recover. And that's what we observe with real-life examples. That's another example. We compare the unpurified mRNA mixed with IVT mixture, so all kind of IVT component. After the elution or after the purification, we could get all of it recovered. And an example of contaminants like an enzyme, which is a typical impurity, there would be that much in the load, so it's in microgram, and we could get everything in the flow through, and what was not in the flow through was below the detection limit in the elution. Again, an example of the recovery, whatever the impurity of the feedstock is. That's just a zoom in of what I showed you before. So we could see that in this, and this results has been made with the samples from AdTech. So Amtech just provided a sample to work with real sample, with real contamination. And you could see here the DNA and enzyme just went into breakthrough, in the flow through, sorry, and we could recover single-stranded mRNA and double-stranded mRNA that we need to separate later. Coming to my last slides, we compare then this technique with this chromatography resin with the existing method on the market. So most of the time the spin column. So in an unpurified IVT mixture, you would get an mRNA with most of the time 15% or above 15% impurity close to the elution peak. With current spin columns, you can reduce a bit this kind of impurities to 13. When you use with exactly the same sample, the chromatography resin oligo DT, you reduce that to 6.2%. And in that case, we can help you to optimize the gradient and everything to separate the two peaks. So as a conclusion, I would say that we have here a chromatography resin which is very specific, that you can scale up, that is very easy to use, no complicated buffers or additives. It's quite straightforward. It can be used with complicated buffers if needed, or if you have specific things to ensure the stability of your mRNA, of course. And it's scalable anyway, as I said. And we are now thinking about the next step. So something that would be as efficient as this one to, for example, separate double-stranded RNA or secondary structures. Yeah. So if you have any question, I'm happy to answer them. And over to you, Stefan. I know I spoke very fast, but I see that the time is over that way.
We're ever so slightly running over time. However, we're going to keep going because we've got loads of questions that have come in that are really, really good. So we are going to try and answer as many questions as possible. Before we do that, though, we have one last poll for you today before we get to Q&A. So this was a knowledge culture webinar. And we also run knowledge culture seminars, which are more one-to-one based, so that your company can select a number of topics, and then we'll organize for a team of Thermo Fisher Scientific experts to give you some of the latest insight on your chosen topics. So the question is, would you like us to get in touch to organize a free virtual seminar with you? The answers, you have the choice of yes, about single-use technologies, yes, about downstream purification, yes, about cell culture, yes, about another topic, or maybe next time, up to you. So would you like to have a technical specialist contact you to schedule a virtual seminar? Okay, I'm going to close that poll off just now, and then we'll move to our Q&A. So, Pirkko, I think we're going to come to you for the first question here. We've got lots of questions to get through. First one for you, Pirkko. Can you mix different peptones together?
Well, thank you, Stephen. That's very good question. Yeah, we do recommend of mixing peptones so that it increases the diversity of the nutrients in the mixture, and you can really fine tune then and find the best conditions for your culture. So yes, the answer is definitely yes.
Brilliant, thank you, Pirkko. Okay, Peter, we have one for yourself here. So this is a question from Claudia, and the question is, what analytical method do you use for poly-A tail length assessment? For you, Peter.
Okay, so we have one, let's say, quick shot method. We can use kind of a PCR method to check for the presence of poly-A tail at all. We can use a certain primer design to amplify the complete poly-A region and to perform capillary electrophoresis to see if this is the expected size. Normally we expect one band, homogeneous signal, and a more detailed analytics can of course be done by mass spec analytics by our external provider. So for us, the first initial QC to show the poly-A tail is there, and there's the biggest part of the poly-A tail is the expected size, can be done via a simple PCR, let's say, and a more defined to see, for instance, if there's a question regarding also a ratio of full size versus less full size can be done by mass spec analytics provider.
Great, thank you, Peter. Peter, sticking with yourself then, I've got another question from you, this time from Amit. Any specific requirement of biosafety level for mRNA-based therapeutic drug production, more specifically self-replicating mRNA-based technology?
A very good question indeed. So the biosafety concerns might occur then in the case when you are producing a full functional or complete viral sequence. We had also requests from customers asking for a complete full genome sequence of a viral. So this would require safety, biosafety levels. Because then there might be a concern that we have an mRNA which includes a full functional viral genome. This is cannot be done without biosafety considerations. But otherwise there are no biosafety constraints at all in that regard.
Okay, Anna, we've got a question here for yourself. Is the breakthrough dependent on the volume of load or amount of product loaded? Does your mass balance closes?
It's more not really the volume of load. Of course, you would not buy or load something like 10 liters on a 1 milliliter column, but that's more the amount which is loaded, the amount that can bind and that is above the nominal DBC of the resin or not. So you cannot bind more than, I mean, what we have seen, that 5 milligram per milliliter is usually a capacity that you can reach, not by everybody, but most of the time. You would not bind more than that, or maybe 6 or 7, but not more than that. So it's more the amount and not the volume. As the contaminants cannot really bind anyway, that will not block the resin. Even if you have a big volume, that will not play such a big role.
Thanks, Anna. I hope that answers the question for our questioner there. Here's one for you, Pirko. Is it challenging to induce heat shock using a single-use bioreactor due to heat transfer limitations?
So the heat transfer in the single use is comparable to a stainless steel reactor, and the diagrams for the both 30-liter and 300-liter single-use fermenter can be provided in demand. And in the study, 1.5 kilowatt temperature control was used to increase the temperature from the 32 degrees to 42 degrees in about 20 minutes. And that was in the 30-liter single-use fermenter. And if you need any more information, do please contact us.
Great advice, thanks, Pirko. Peter, we've got another question for yourself, this time from Benjamina. Does scale-up come with a significant cost reduction?
Could you perhaps please repeat the question, Stephen? I didn't get it properly.
The question is, does scale-up come with a significant cost reduction?
That's a very good question. Of course, scalability, that's certainly the expectation of customers, that scalability has also an impact on price. Yes, certainly, we will see, and since we have discussed that the methodology is very simple, very easy to implement for scalability, this will definitely have also an impact on pricing. But I think this time I would not like to focus on any price discussions, but it will have an impact on pricing for sure.
Right, thanks, Peter. Anna, back to yourself for another question. You said one loads with salt and wash with salt, then you looked in water. What salt concentration are we talking about?
So usually you would start around 0.5 molar salt, so NaCl or KCl. That's quite a typical one. You can adapt it to the stability of your mRNA, of course. Then you would probably wash with quite less, but still a bit, like 100 millimolar, and then elute without salt at all. So you could stay with a buffer without salt or elute with water. And you will also regenerate with water before cleaning the resin. It's quite straightforward.
Okay, thank you. Sticking with yourself then, Anna, this customer is quite happy with the reversed phase method. They're asking, why would I switch to your Oligo DT?
It's not that empirical compared to reverse phase. You can use a lot of additives, a lot of organic solvents. It's a quite easy one. You can go very fast with the flow, and it's very much scalable. So I think, and it's very specific too. So you would not bind that many things that are not containing poly-A tail. It's quite selective, and yeah, I would say that you would gain in time in recovery, yield, and scalability.
Okay, thank you. Peter, I think we'll come back to yourself then. The question is, does the size of a synthesized mRNA impact on purification challenges and stability of the molecule?
Yeah, that's a good question. So maybe getting started with stability. So stability is, generally speaking, not only a matter of size. It's more or less a matter of sequence. We have seen that some also very short mRNAs were not as stable as very long mRNAs. So this is not really predictable. So generally speaking, the long mRNA is not automatically less stable. And purification, of course, a very long mRNA has an advantage to get rid of low molecular weight products. So this because there is a big distance between a long full-size product and small-size product. Just to give an example, if you have very short mRNAs and you might have both products, this is sometimes they are near almost in the same region. If it happens that the sequence is problematic, for instance. So I think it's not a problem. It's more even an advantage to purify longer mRNAs. And stability is, as I said, not a matter of the length in general.
Brilliant. Thank you, Peter. And I think that's a good place for us to end this webinar today. So thank you for sticking with us. I know we went slightly over time there. We had so many questions. What we will do is we will endeavour to answer those questions and send the answers to the people that asked those questions as well. So we're going to close the webinar there. Thank you so much for your time, and we really hope you got something out of this webinar. Please feel free to sign up to the one next month on the 22nd of October, and we'd be very grateful if you could also fill in the survey at the end of the webinar as well. Thank you so much, and have a great day.